In previous Health Tree University lessons, we explored how gene mutations are classified. Now, let's zoom out and focus on the bigger picture, the chromosome. In this lesson, we'll dive into the different types of chromosomal mutations and how they can impact our DNA. Get ready to unravel the next layer of genetic changes. What are the different types of chromosomal mutations?
When we look at genes and cytogenetics in multiple myeloma and the chromosomes of the cancer cells, we try to look to see if there's any mutations. And there's different types of mutations that we can see in cancer. There's translocations, there's deletions, there's insertions, there's amplifications, and each one means something completely different. So let's start with defining each one of these. So deletion is where a section of the chromosome in that cancer cell is removed or lost, and it's disappeared completely. And whenever we say that, we normally refer to it as a deletion of the chromosome and a specific portion of the chromosome and the majority of the times. So a deletion of 13 chromosome or 13q deletion or deletion of 17p. Those are just examples of deletions of parts of the chromosome that are related to multiple myeloma. Now, for example, in deletion 17p, we're talking specifically of a small portion of the chromosome number 17 that is part of the 23 chromosomes that are in pairs in the cancer cells. So for 13 deletion, the whole 13 chromosome is missing in the cancer cell. Sometimes you can have the whole chromosome missing, like 13 deletion, or you can have fragment of that chromosome missing, like the 13q deletion. There's other types of chromosomal mutations, like translocations. And translocations is when a section of the chromosome is transferred to another chromosome within that same cell. So an example of that is translocation 1114 or translocation 414. That means that there's a portion of the chromosome 4 that moves to chromosome 14 and portion of the chromosome 14 that moves to chromosome 4. And they shouldn't be there in a normal cell. So that, for example, is a translocation 414. And just like that, there's several translocations in multiple myeloma that we look at that can help us establish whether a patient has high-risk disease and likely to relapse or if they have standard disease. Now, there's other types of mutations like duplications. What that means is that a part of a section of the chromosome breaks off and has more pieces added to that same part. So it's basically reproducing or replaying the same gene within a chromosome. And that causes extra genetic material in the gene and in the abnormal cell. There's also insertions where there's a portion of a gene that was either floating around or that came from a virus or from a foreign portion or within the same genome of the human cell that actually gets inserted into a chromosome without actually having it being replaced with another, like the translocations. And the insertion, instead of switching places, is just basically adding one extra gene or portion of the chromosome to an existing chromosome. And inversion is another type of mutation that we see where a part of the gene within the chromosome breaks off, flips, and then gets fused back into the chromosome. So basically the material that it contains is all inverted. And it's as if we were reading backwards the recipe of how the cell should grow and how it should behave. So those are the most common types of chromosomal mutations. In multiple myeloma, the most common ones, I would say, are deletions, translocations, and we also see an excess amount of chromosomes, hyperdiploid, as we use it in medical terms, or a loss of the complete chromosome, which we also call hypodiploid, or a lower amount of chromosomes than the normal 46 chromosomes that we have.
To learn more about hyperdiploidy and hypodiploidy and how these chromosomal changes affect outcomes, make sure you watch the video on this topic in Health Tree's Chromosomal and Gene Abnormality course. Understanding the genetics of your cancer is crucial. Knowing your risk status can help guide your decisions about treatment and maintenance therapy, and it may also reveal a targetable mutation. To track your genetic profile, create a health tree account and connect your medical records. You can view your genetic profile by clicking the Track My Disease button on your dashboard. The health tree algorithm will then use this information to suggest treatment options and relevant clinical trials. Thanks for watching.